Welcome to Decoding Healthcare Research. I'm your host, Efrain Riveros, Dr. E.F. Rain. Today, we will discuss the microbiome. How is it important in the context of aging? This is a topic that is not only interesting, but important to the well-being of every human being. And I think is very pertinent to start discussing this with an expert. We have the honor of having here with us Dr. Filipe Cabreiro. He's a professor at the University of Cologne and the Imperial College of London in the UK. Welcome, Dr. Cabreiro.
Thank you. My pleasure.
Excellent. So we are going to start discussing the relationship between the microbiome and aging. This is based on a review published in the Annual Review of Genetics in the year 2019. And I'm going to start by asking Dr. Cabreiro, what sparked your interest in this topic?
That's a good question. It came a bit by chance, actually. So I've been working on the aging field since my PhD. Actually, I started working on cell biology of aging, looking at how antioxidants and antioxidant enzymes regulate damage in cells and its potential link with aging. And then I started moving to a postdoc in the UK, where I worked with David James at University College London. And at that point, we were using this small nematode worm as a model, which is a very good model for studying aging, and it's led to very important discoveries in the aging field. And we had an interest in trying to already use compounds and drugs that could potentially improve the process or the aging process. And one of those drugs that we were looking into was a drug called Metformin, and Metformin is a type 2 diabetic therapy. And when we tested it with our model, we actually realized that the positive effects of the drug were mostly due, at least in the longevity context, were mostly due to its impact with the gut microbes. So that really sparked a lot of interest at the time, both in our side when we were doing this research. Now everyone talks about the microbiome, but this 15 years ago, there was very little interest and knowledge actually on this topic. So we were one of the first people actually linking how drugs can impact longevity, actually acting through microbes. And this was then shown in other models to be conserved. So we were quite happy to be part of that original discoveries of drugs and microbiome and aging. So that kind of sparked the interest. And then I started my own lab, both first at UCL, and then I moved to Imperial to continue that line of research where we looked into the effects of different compounds, but also microbes themselves on different parameters of aging.
Yeah, and aging is central to many things, and many actually illnesses have intersections with the process of aging. And you start your paper by mentioning a framework that was initially published by Dr. Lopez Ortin in 2013. And he talks about several things that happen with aging, some of them directly related to epigenetics and the genome itself, but also some dysfunctions within the cell. I think as a starting point, we can refer a little bit to this framework. What can you tell us specifically about it?
So this is a very important paper by Lopez Ortin on what they call the hallmarks of aging, and they looked at the literature and tried to find what are the most important factors that may underlie or explain why we age. And they did find indeed that epigenetics, DNA damage, proteostasis, so the inability of the cells to maintain a proper folding of proteins so that the function of the cells and its proteins work properly. So that all the case with aging. But there are other important phenomena or hallmarks, such as dysregulated insulin signaling, or mitochondrial homeostasis, so mitochondrial being the kind of the powerhouse of the cell and the ability to produce energy. It is very important to maintain these mitochondria in a good homeostatic state, so they continue to produce ATP or energy for the cells without overproducing reactive oxygen species, which can be damaging. So all those kind of came up as being very clearly mechanisms that we can identify in many different aging processes or parameters as being essential for the decay of a cell or an organism. And what we proposed based on that review that you're mentioning is this idea that we have that actually there are other hallmarks. And interestingly enough, so we suggested at that point that the ability of the microbial communities to maintain that homeostasis is equally important. And we do observe that during the aging process, the microbiome that we, when we're healthy, starts changing and it starts becoming more unique. In some cases, it becomes so, what do we call this, biosis, so different that it can actually lead to disease on its own. So, and interestingly enough, Filipe then did an update in 2021, if I'm not mistaken, on the hallmarks of aging, and it then included, as we did, proposed already in 2019, dysbiosis as a new hallmark of aging. What is so striking, actually, is that one can, even just by looking at the microbiome, one can, through computational methods and AI, actually predict human aging. So it really is a clock that is aging as we age and can tell us how old we are. There's an error about 5 plus minus 5 years, but it's still quite remarkable that just by looking at the microbes that we carry in our gut, we can actually tell our own age. And I think this, again, really supports this idea that we are kind of all aging as a meta-organism rather than as a human and a microbial component. So we're all kind of this concept of what we call the holobiont, that we are more than just our own cells. And this is really, I think, quite well encapsulated even within the aging context.
Yeah, and this perspective is actually a complete change in paradigm, and I think the incorporation of the microbiome and into the perspective of just the human genome is important. And my question is, you say that as you age, it's not only you that are aging, the microbiome is changing as well, and they go hand in hand. And you talk about the hologenome. Can you elaborate a little bit more about that concept of the
Absolutely.
hologenome?
So the hologenome was actually coined originally by the Rosenbergs, and this is the idea that our genome is usually fixed. We can't add more genes, but the microbiome carries 100 times more genes than our own genome. And that gives us an array of possibilities that our own genome does not. And one of the most simple ones, the most easily explained actually, is our ability to obtain energy from fiber. So we as humans, we do not carry the enzymes that can degrade fibers. But by carrying these microbes, they do possess the enzymes, they do possess the machinery that can transform fibers into short-chain fatty acids. And these short-chain fatty acids then become energy sources for our own cells. So they can then travel to the bloodstream, or even act locally on intestinal cells and provide energy directly. So it really shows this concept that we're really more than the sum of our own genes. We're really the sum of our own genes and the microbes that we carry. And that gives us the ability to adapt to a constantly evolving environment. And the hologenome theory came from that throughout evolution, we acquired these microbes, which gave us the ability to quickly adapt to a constantly changing environment. That could be from the things that we eat, but the things that we're constantly exposed to. And this really gives us this buffer to quickly adapt, which we won't be able to by just simply our own genes. And I think this is what we think is probably very important, both in the science context, but also in the medical context, that we need to look at the whole thing together rather than as separate parts.
Yeah, and the microbiome is getting more attention by the day. And as the microbiome changes as you age, there must be mechanisms. So let's go to the pathophysiological mechanisms, what happens as you age? What happens with your microbiome, and what are the molecular interactions there?
So one of the things that does happen is the one theory of aging is also the inflammaging, which I think goes beyond just being a theory. It's this ability of our bodies to no longer contain and maintain a level of inflammation. And of course, inflammation is also important. So it's kind of a double-edged sword here. But we lose the ability to keep an homeostatic level of inflammation, and this then leads to, for example, a loss of barrier integrity in the intestine, and this allows the translocation then of molecules from microbes that do then cause enhanced inflammation, and you end up in this vicious circle or cycle of negative feedback of inflammation that just keeps going on. And indeed, it's been shown that in animal models, that in particular mice and rats, that if you do not carry any microbes, you can dampen quite dramatically that inflammation. And indeed, some of those animals do live even longer than animals that do have a microbiome. What is also interesting is actually, as we discussed before, the microbiome is also shifting and changing, and this has to do with many things, many factors, some external, some internal, inflammation being one of the internal ones. But we do start shifting towards microbes that tend to have a more kind of pathological, this idea of pathobionts. So when we're healthy, we can keep most of the microbes that are more detrimental kind of in check. And as we age, we lose the ability to contain these microbes in that state, which allows then, of course, to have a more kind of pathogenic role of those microbes to pervade as we get older. So we kind of always working towards a situation that is far from optimal by having a dysbiotic microbiome that produces more molecules that are detrimental, and at the same time our cells are less capable of keeping the integrity and the ability to counteract those negative effects. So it kind of goes together.
So we may say that as you age, you'll start losing diversity, and the result of that diversity is also like an imbalance, right, between
Absolutely.
good and bad microbes. And you mentioned that inflammation is central, and gut permeability is probably part of that. How is that inflammation connected to metabolic changes that happen as we age?
So this is not entirely my main area of expertise, but my one of the things that the signaling pathways basically that are involved in keeping check inflammation, they do interact with the insulin signaling pathway, and that leads to then the inability to maintain like glucose homeostatic levels, etc. So there's many ways about how the genes and the pathways that are involved in controlling inflammation can have kind of local effects on cells by acting with other pathways that are involved in glucose, but also lipid homeostasis, et cetera, et cetera. So it's quite a big field there, and I have to say it's not entirely my main area of expertise.
I see. So when we talk about diet and nutrition, one of the aspects that people usually go to is calorie restriction. And I would like to ask you, in the context of your research about metformin and let's say calorie restriction, they share some commonalities. So let's talk about metformin specifically, so that we can illustrate what happens with the calorie restriction.
Yeah. So metformin is one of those molecules that, interestingly enough, right now probably wouldn't be approved by the FDA, because we still, after 70 years of its use, we still do not know exactly what the target of metformin is. Of course, there's a lot of research saying it's complex 1, but there's which is an important membrane protein in the mitochondria that is involved in the production of ATP. But it's still not entirely clear that it is the only target. So there's the literature is incredibly extensive about the many different pathways that metformin acts on. We've written a review on that in 2014, and pretty much every month there is a new mechanism, a new way of how metformin works. So our work did show that by acting on bacterial folate metabolism, this could have an impact on how the host physiology would be changed with the treatment of metformin. So metformin is one of those interesting molecules that by doing that, it activates AMPK, which is a central regulator of energy in the cell, and it does improve then the balance between catabolism and anabolism, which is important for the cells, and hence the kind of caloric restriction phenotype that metformin seems to induce. So it's interesting that there was an interesting paper from Frederick Backhead in 2017 where they treated mice they treated patients with metformin, and they took the microbiome of these patients before and after treatment, and then introduced just that microbiome that had been from those patients before and after treatment into naive mice. And they could see that actually the mice that had received the microbiome from the patients that had been treated, so after treatment, had a much better hyperglycemia control of glucose homeostasis than the mice that before treatment, really showing this ability that metformin has direct effects on the cells, but also indirect effects by going through the microbiome, by changes in the microbiome, and those can also be positive towards an improved glucose homeostatic effect, basically.
So that's very interesting, and it, as I mentioned before, there are some intersections and commonalities with calorie restriction. And you mentioned some specific metabolic pathways. You already touched tangentially on AMPK, but you also talk about the effect on insulin and IGF-1, as well as the effect on mTOR. And basically transcription and translation into proteins. Can you integrate that a little bit better for us?
Yes. So they all kind of play with each other, and the pathways are individually. If we target each one of them genetically, we can, for example, improve cellular homeostasis, but also longevity, actually. So this ability to kind of dampen the pathways, both by deleting, for example, the insulin signaling receptor, or by shutting down mTOR, anything that kind of continuously produces more energy or proteins, or that if we can stop that, we can improve basically cellular homeostasis, and that really leads always pretty much to longevity. But they don't act just only alone, that we can do genetically, but often when we use through caloric restriction or through the use of drugs, they tend to play with each other. So there is kind of really an interplay between the different signaling pathways, and caloric restriction is a clear example of that, where pretty much all these pathways get engaged, right? So there are, and then depending on the caloric restriction method, some will be more dependent on FOXO, which is the transcription factor under the insulin signaling pathway that regulates the transcriptional changes in the cell that are important for the metabolic adaptations that are needed for those positive effects. But some of them will be much more going through mTOR, which will be then regulating a different subset of genes that are also important for cellular growth and also metabolic homeostasis, or AMPK, which is really the kind of the homeostatic rheostat of cellular balance, basically, of energy balance between AMP and ATP. So it really senses AMP and ATP ratio, and it then shifts between anabolism to catabolism, or the other way around, depending on the demands of the cells. So caloric restriction really plays upon all these different things. And the beauty with caloric restriction, at least, and the promise of it, is that it's been tested in many organisms. It works really well in yeast, it works well in C. elegans, it works well in flies, it works well in mice and rats. In monkeys, there's still some discussion around it, whether the animals were really at a normal level of food, or whether they were being overfed, and that's why caloric restriction works. So there's some discussions there that are still debated. In humans, there's still, of course, we do know that caloric restriction helps with a lot of parameters. Whether that will lead to a lifespan extension in humans, there's no evidence at the moment for that.
Yeah, but it's exciting that the research is moving in that direction because this is something that becomes practical for people. So knowing that having calorie restriction is having all these effects that even though they are difficult to isolate, you know that it's attacking the problem from different corners, and that's actually exciting. And I want to go back to the original framework. And one of the factors that are affected during aging, during the aging process, is the homeostasis of proteins or proteostasis. So let's focus on that. And what are your thoughts about that particular aspect?
So I think it's an interesting hallmark, and it's almost, as you say, if you compare proteostasis and metabolism, they kind of tend to be there's not a lot of overlap between the two. There is some evidence that there's communication between the two things, but not so much. We did, though, publish a paper in 2021 or 2022 where we did show that if we have a ribosome, a subunit of the ribosome that can make proteins with fewer errors, that alone. So this was an interesting study where we actually looked at ribosomes from different organisms, including archaea. So archaea are kind of evolutionarily very distant from us, but we started looking at different archaea and different organisms, and we did find that the ones that lived in very harsh environments, and the reason behind this was we were looking for how do you make proteins when the conditions are so harsh, right? And some archaea live in very demanding temperatures, which makes, of course, with temperature, proteins tend to unfold. And we wanted to really understand how can you make proteins of high quality in such demanding environments. And then by doing a computational study and analysis, we did find that there was a single mutation in one gene in a ribosome that seemed to be only conserved in these hyperthermophilic archaea, but not in the normophilic ones. And we said, okay, well, this is very interesting. And then we picked that single mutation, which didn't exist in eukaryotes, and we changed that hyperthermophilic archaea mutation and we changed the genome of both yeast, C. elegans, and flies. What was really striking is that by just changing that little single nucleotide polymorphism, basically, by doing that change, we were capable of now creating animals that were more resistant to heat stress. They were more long-lived, robustly long-lived, and had very minor, actually, negative or side effects, which was they were just a bit slightly delayed in development. But the gain in terms of health, or especially adult health, was quite striking. So that really told us that indeed this ability to be able to produce proteins, and we followed it up with a lot of molecular studies as well in this paper, that we could create proteins with few errors as the animals were getting old. So as we get old, our ability to produce proteins loses the capacity to produce perfect proteins with time. This idea of fidelity, of protein fidelity, and those then if we can't make proteins that are properly folded, then that leads then to the cells need to get rid of them. But sometimes the ability of our cells to get rid of misfolded proteins is lost with time as well, which makes then a big problem for the cells. And we did realize that indeed, if we can change the machinery that produces this protein to make them maintain this fidelity, the quality of these proteins over time, that alone is sufficient to actually improve our resistance to thermal stress, but also our ability to live longer. And this was quite an interesting finding, which is picked up. What we did find then, and this is what I think it's interesting to kind of link maybe with metabolism, is that an mTOR seems to be the kind of probably the regulator that links a bit both, because it does have a role in metabolism, but also in protein production. And we did find that rapamycin, which is one of these mTOR inhibitors, did not longer extend longevity further. So rapamycin, when given to model organs, extends longevity, improves health. But when we gave them to these organisms that had already that mutation, it did not extend further, really showing that part of the mechanisms of rapamycin are not just metabolic, but also partly dependent on improving the quality of the proteins that are actually being produced over time. So rapamycin really does play both on a metabolic angle, but also in the context of improving fidelity of the proteins that we produce. So hence the importance of proteostasis for an aging organism.
Yeah, it's clear that the synthesis of proteins and proteins of good quality is essential for longevity. Let's try to go closer to the nucleus. So we have been talking about the proteins. Now let's talk about the DNA. I remember when I was in college, aging was a synonym of telomere shortening. So that's something I learned when I was young. But it definitely goes well beyond that. And you mentioned actually the telomeres, but you also talk about changes in DNA and chromatin, the methylation processes, and also histone alterations. So tell us a little bit more about the DNA in the process of aging.
So that's another kind of big area of research. I have a colleague actually in Cologne who's an expert on this, Björn Schumacher. And indeed, as we age, again, the ability of our cells to maintain our DNA without errors is lost over time. The replication of cells loses the ability to maintain those. So there are several processes that try to keep in check, and that there's not an accumulation of mutations over time. But those, again, with time, they do lose the ability to maintain them. And again, if we have a genetic code that is coding for incorrect proteins, then that leads, of course, to several issues, including proteostatic issues as well. So there's a very nice connection there. The idea of telomeres is one of those, I think it's indisputable, the importance of telomeres during aging, especially human aging. And yet, I always think about the example of C. elegans, who doesn't have telomeres and still ages, right? So it is one of the important components, and I guess indeed for more complex organisms, it plays a key role. But it's not the explanation that people tried to give when it was mainly there was a big hype at the moment that it would explain everything, and I don't think it stood the challenge of time. So I think it is, again, a contributor, but not the sole thing that we should be working on. The other point that you mentioned about is methylation, and methylation is a very interesting. It's being so as we age, there are definitely changes in our epigenome as marks that tell us, again, very neatly what our age is. So there's very nice work where by just looking at the epigenome, one can get to a very accurate biological clock, actually, not chronological clock of aging. And that tells us really whether let's say if I'm 43, what is my biological clock? If I do an intervention, if I start running, if I start doing caloric restriction, is my biological clock going to improve or worsen if I start eating, processed foods or very unhealthy diets? So I think we can use the epigenome a little bit like, as I mentioned earlier in this conversation. We can actually use these parameters like the dysbiosis of the microbiome that can tell us our age. Well, we can also very, very nicely actually use the epigenome clocks to tell us our biological age. And I think they're becoming a very important tool, I think, also for the medical world to really assess someone's age and devise a very clear strategy, almost in a personalized way, to check someone's ability to improve with time. So I think it's almost of course, in terms of how is this going to happen in terms of insurances, et cetera, there's a big discussion going on there as well. But I think from a medical and personal point of view, I think it is a fantastic tool to have, to be able to look at our epigenome and to tell us and to work on a plan to make things better.
Yeah, one of the aspects that I am taking from this conversation is that multiple things happen at the same time as you age, and aging is a multifactorial process. But the role of the microbiome in this process is critical, and it changes as you age, and multiple metabolic and genetic changes happen at the same time. So to close this conversation, how would you, in a nutshell, try to summarize the content of this paper for a layperson?
So I think it's as you said, and very well, this is multifactorial. There's actually a large discussion right now going on about whether we should even classify aging as a disease, and that would change a lot of things. There's a lot of back and forth on this, and there's a lot of pushback as well. It's definitely the mother of all diseases, that's for sure. Whether it's a disease in itself, that could be just semantics or something else. But that would change also this idea that maybe we could potentially treat aging, right? So with the idea with rapamycin, metformin, and other therapies, you could go after the process itself and improve all these different diseases. So my take on all of these is we should not, again, when looking at how we treat aging, we need to take into account not only the different hallmarks, but also, as we said earlier, this idea of the hologenome and the holobiont. We need to treat the entire organism, not just the host cells. And I think if we do this, or even explore the ability of our microbiomes to even produce molecules of interest, then we should be able to enhance human health in a much more predictable way as well. Because right now there's still a lot to be understood and known from the microbiome side. As you said, and very, very well, it does touch upon pretty much every single process of the human body. And yet we do still know very little about how does the microbiome actually communicate with the host. And we often talk about short-chain fatty acids. We talk about indoles, which are another class of molecules produced by the microbiome. But the microbiome basically contributes with almost 60% of the entire content of molecules that we constantly have in our bloodstream. And we know very little about how and which microbes and in which context they produce those molecules of interest. And I think that the research needs to go a lot more in that direction to understand those mechanisms and put them into the context of this massive multifactorial process that is aging. And I think this is a big challenge, of course, but it's something we should work towards.
Yeah, that's absolutely true. And thank you so much, not only for giving us that perspective of, this is probably the first step to go deeper into the role of the microbiome. And I am sure as the research progresses, we are going to find new insights that are going to help us stay healthier. And in particular, the research that you are doing is very enlightening. And we appreciate you sharing this information with us. And we will follow up on your research. And I hope in the future we can talk again to see where things are going. I want to thank you for being with us here today, Dr. Cabreiro. It has been really a pleasure for me, and I'm sure our audience is going to enjoy a lot of what we discussed here. Thank you so much.
Thank you very much. It's been an absolute pleasure. Thank you.
Thank you. And for our audience, you will find the link of the paper in the description of the video and some details about Dr. Cabreiro and his lab. See you in the next episode.